show some highlights of my research in the Laboratory of Ecological Chemistry of Hokkaido University. Hokkaido University, I first landed Hokkaido Island, very close to Siberia in 1997. And it was, big surprise, huge snow in Bangladesh. We have never imagined how much snow there. But my topic was very interesting: signaling and communications between plant and peronospore Oomycete, phytopathogen, chemical ecology, how pathogen and host talk with each other through small signaling molecules, especially secondary metabolites. When I visited the Professor Satoshi Tahara's lab, he is now 81 years old. Recently I visited Hokkaido and he offered a social gathering. So his lab was looking at the how zoospore of Aphanomyces cochlioides, a pathogen of horensu, that is spinach and sugar beet, find the host, and then zoospore, shed flagella, becomes cystospore and infect the plant. And this pathogen, Aphanomyces, has limited host range, only few members of Canopodaceae and Amaranthaceae. So he gave me the target to discover the chemotactic signal and then how zoospore perceives the signal and moves to the cystospore and life cycle continues. So it was very exciting, job for me. But first, I saw that when a root tip of sugar beet or spinach you drop into the zoospore suspension in a petri dish, within a minute all zoospore aggregate. But zoospore I cannot see here, only a small dot under light microscope, because 5 to 6 micrometer in size. So and we did a lot of work to finally find the specific, host-specific chemical signal, cochliophylline A, and that one we published in the journal. But most exciting thing is that here in the microscope, zoospore is just like a dot. How this dot moves and find the host, it was another target. I moved another direction to study the structure of the zoospore. So recently I visited the technician who trained me to use the scanning electron microscope and transmission electron microscope to fully understand the complex structure of the zoospore. The zoospore has two flagella, one anterior, another posterior. Anterior is used for swimming and this one is steering. So this was very fascinating journey at that lab. And then we asked question whether cochliophylline A, when zoospore reach to the host surface at higher concentration, can trigger differentiation, that means becoming cystospore and germination. And yes, we got very fantastic result that at higher concentration of cochliophylline A, 10 to the power minus 8 molar concentration, you can imagine how low concentration, less than nanomolar, this signaling compound works. Yes, it can assist and cause germination. And then we asked a question: how zoospore morphologically dramatically change? Dramatic cytoskeletal changes taken place, and we studied this molecule and visualized the actin and tubulin, two cytoskeleton. But we found that this cross kingdom molecule can trigger polymerization and depolymerization of the F-actin in the cell. Really amazing study. I had very fantastic time over there. And we also studied the receptor in the zoospore membrane. We found that, I am not showing the in detail, G protein couple receptor present in the zoospore membrane perceive the cochliophylline A signal and then downstream it through phospholipase D and production of the phosphatidic acid and calcium second messenger. So this is a story about the host pathogen interaction. But Tahara Sensei asked me to check why other plants are not host of the Aphanomyces cochlioides. So this is a vegetable I brought from Bangladesh, but this should be the host of Aphanomyces cochlioides. but we found 100% resistant. And then we found in the extract, two compounds are there. This nicotinamide can halt and stop the zoospore, not allow to approach the root. And we finally made this conclusion. In case of non-host, nicotinamide can induce encystment, but zoospore cannot germinate, produce again zoospore and 3 cycle, then it is ending. But in case of host, like this way. But this fascinating compound, cochliophilin A, only been found in the host of Aphanomyces cochlioides. No other report yet. So this we discovered lots of compound. I am not going in detail. We also found some compound in the bacteria, rhizosphere bacteria, and we also discovered some compound. So even when I was in Germany, we also studied another Oomycete, Plasmopara viticola, and discovered lots of compound. And one of the compounds, taurosporin, we found the mechanism is inhibition of the protein kinase C in the zoospore cell and stop the motility and control the disease. So this is all about my some highlights of the past research.